Good morning, everyone. Today I will talk about microbial conjugation. So, conjugation is a technique of gene transfer. Bacterial conjugation was first discovered by Lederberg and Tatum in 1946 in ECHS coli 12 strains. Now, conjugation transfer of genetic material from bacteria A to bacteria B by mating or contact is called conjugation. So you can see here in the diagram, two bacteria are shown, bacteria A and bacteria 2. So a pili is formed, and with this there will be the transfer of genetic material from A and from bacteria A to bacteria B. So this process is called conjugation. Now, who will be the donor? F plasmid donor, F plus male cell, and absent receptor F negative is female. So donor is the that cell from where the genetic material will be transferred. So which is F plasmid. So now S plasmid, fertility factor, sex factor, and transfer factor. The F plasmid encodes for the sex pilus. See, in F negative cell, and this is F positive cell. With the help of this sex pilus, the genetic material is transferred from one to the another. What happens during conjugation? So discussing about the different steps that are involved. See here, donor cell attached to the recipient cell with its pilus, that is known as conjugation pilus. The pilus draws the cell together. See, as shown in the figure also, there is a F plasmid. So with the help of this conjugation pilus, this F plasmid will be transferred to the another. The second step is the cell contact with one another. So with this conjugation pilus, the two different cells will contact to each other. In the next step, one strand of plasmid DNA transferred to the recipient. See, like in the third step, as shown in the diagram, the one strand of plasmid DNA is transferred, and after that, the recipient synthesis a complementary strand to become an F positive. The donor synthesizes a complementary strand, restoring its complete plasma. So what happened in the third step? A single strand of plasmid DNA was transferred to the another side, and then in the next step both the cells became F positive, as in case of donor 1 the plasmid restored its complete plasmid. Now the once female recipient cell now becomes the male donor cell, because the plasmid transferred from one to the another. This characteristics of maleness in bacteria is transmissible for infection. Now, what is HFR cell? When F factor plasmid exists in integrated state with the host chromosome, it is called HFR cell. F plasmid integrate into chromosome by recombining. Now, talking about HFR conjugation, you can see here in the diagram, F plasmid integrates into chromosome by recombination. Then cell join via conjugation pilus. After that, portion of F plasmid partially moves into recipient cell, tailing a strand of donor's DNA. After that, the conjugation ends with pieces of F plasmid and donor DNA in recipient cell. Cell synthesize complementary DNA strand. Donor DNA and recipient DNA recombine, making a recombinant F cell. Now, the F positive free plasmid is irreversible to HFR integrated. F positive free plasmid is turns A into turns F negative into F positive upon conjugation, and HFR conjugation with a HFR and F negative relay becomes F positive because as the plasmid is transferred but it received chromosomal DNA from the donor. Now as you can see here, F positive plasmid was there and then it is reversible from HFR. When the F factor reverts from integrated free state, it may sometimes carry with it some chromosomal DNA from the adjacent side of its attachment. See, as you can see here in the figure, HFR, then excision, after that F negative, then conjugation of P and F cells, and finally the individual F and P cells are formed. So such an F factor is known as F factor. Now F dash plus F negative equals to sexduction. So it means when F dash conjugates with the recipient F negative, it transfers along with the F factor the host DNA incorporated with it. As shown in the figure, HFR equals to excision, then F dash plus F negative equals to conjugation of F dash and F cells, then the two individual cells are formed, F dash and P. Now, medically important factors which can be transferred by conjugation are COL and RTF. COL is cholinergic factor and RTF is resistance transfer factor. So these two factors are very much important medical factors which can be transferred by means of the conjugation. Hope you understood about today's presentation about conjugation. Thank you.